Welcome back for the last session before the closing session. Now we are going to present the winner of the Best Paper Award last year in Cáceres. The recipient of the Best Outstanding Paper Award 2022 is Carlos Alberto Ramos Guzmán from Universitat Jaume I with a paper on the mechanism of SARS-CoV-2 main proteins with ketone-based inhibitors unveiled by multi-scale simulations. So whenever you're ready, Carlos.
Hi, good afternoon. Thank you, Alberto, for your kind introduction. So today, I'm going to show you the work we published in Angewandte Chemie about COVID-19, which led us to receive the Outstanding Paper Award in 2022. This work was made at the University of Valencia while I was doing my doctoral studies, and I did this under the supervision of Iñaki Tuñón. So, while I was doing my doctoral studies, I was working in an enzyme that is related with the Alzheimer's disease. And by that moment, the COVID-19 hits the world. As we were working with the cysteine proteasa, we realized that the experience that we gained with the Alzheimer's disease will be used here. So since then, we center all our efforts in trying to understand the molecular mechanism of the enzyme that is involved in the coronavirus to spread the infection. So, okay, all of us know how the disease is spread. All of us knows that coronavirus is an airborne transmitted disease. That means that the particles, virus, need to reach the surface of the cell in our lungs. When that happens, a fusion process between the membrane of the virus and the membrane of the cell occurs, and after that, the RNA of the virus is released, which is translated by the ribosome in the cell into two non-structural polyproteins will later be cut as they can be used in this way. So these two big polyproteins contain two small enzymes named the 3CL-Pro, or main protease of the coronavirus, and the papain-like protein are those that you can see here in yellow and in white. They perform most of the cleavage events that these big polyproteins need in order to work. Once they are cut, they create the machinery the virus needs in order to continue this replication process. So basically, after this machinery is formed, occur the subgenomic replication and the genomic replication that will lead to the production of the raw materials needed to ensemble more virion particles. Once the raw materials are created, our cells are in charge to create more virion structures. And once our cells create those structures, they are released outside of the cell with the end of continue the spread of the infection. So basically, by observing this mechanism, we can see that if we can inhibit the main protease of the coronavirus, we will be able to stop the entire replication cycle of the virus. The previous was an equal point of view of the problem, but now we need to start to go inside, to go deeper into the problem, because we need to see exactly how can we stop this mechanism. So the first thing that happened is that the substrate binds to the enzyme and create a non-covalent complex that is called Michaelis complex. Once this Michaelis complex is formed, that bond that I'm showing in dotted lines is cut. And after this bond is cut, the first product of the reaction is released. And here by the addition of a water molecule that in the media, the second product is released. What I'm saying is that basically we can say that the reason why the virus continues its replication process is because this peptide bond between the carbonyl and the nitrogen is being broken. So we need to envisage a competitive inhibition strategy in order to avoid this happen. So how can we do this? So if we are able to design a molecule that be able to join to the enzyme faster and stronger than the substrate, in that way we are going to avoid the Michaelis complex be formed. And if we do that, so the proteins will stop to being produced, and then we will stop the replication cycle. So far, we know how the enzyme is reacting with its natural substrate. We can see the natural substrate, or at least a small portion of it, at the top of the slide, and you can see that there are some colors with some P groups. The P groups that are at left of the P1 group are the ones that's going to be bonded during the first part of the chemical reaction to the enzyme, and the ones that are at right are the first that's going to be released. So, how can we do something that is stronger than the natural substrate? So we need to define something that we call warheads, in order to trick the enzyme to react with them, thinking that we are like giving the substrate, but when it reacts with the enzyme, it will form a more covalent and more stable complex. In this way, the enzyme will be stopped, will be stop his replication cycles. One of these warheads is a really one that is being used are these nitriles. When the enzyme reacts with this nitrile, it will form an thioimidate. That is a really strong covalent bond product, and from this, the enzyme will be inhibited. One example of this is the molecule that is contained in the drug Paxlovid. This is the molecule that is given nowadays in order to stop the spread of the infection of the people that acquire the COVID-19. Another one example of these warheads are the mycoreceptors. These form thioethers that are also highly stable, and one example of this is one inhibitor that is called N3, which is a potent inhibitor with that has shown to be very effective against different coronavirus cell-based assays. And finally, aldehyde ketones forms methioketals, and this is the kind of inhibitors that we selected for this study. As I told you, we need to create something that be able to trick the enzyme. As you can see here, in the P1 position, there is a glutamine. And what we are doing here is our inhibitor, we are using a structure that is very similar. The main difference is that now the glutamine is creating, now we are creating a cycle. This is called a gamma-lactamine ring. And by including this gamma-lactamine ring or creating this gamma-lactamine ring, we are decreasing the conformational freedom this side chain has in the enzyme of the enzyme. So, and the same strategy is followed for all the other groups. So, but how can we use computational methods in order to try to stop, or at least trying to explain how a disease is being communicated or spread? So, for using these computational measures, we can evaluate at an atomistic level how the energy is changing while the reaction is occurring. And this is something that can be done using non-computational methods or any other kind of experimental methods. So we can obtain the entire path of the chemical reaction, and by using the transition state theory, we can relate one computational measurement with one structure, with one experimental one. That is the catalytic constant. This catalytic constant is giving us information about the speed of the reaction of the enzyme with the inhibitor. So if we get a higher barrier, so the energy will be it will not be fast enough in order to react with the enzyme. Experimentally, what we can measure are the values that I'm showing here in red, because basically what we are doing in the lab is we are mixing reactants and we are measuring how fast they are consumed, but we are not able to see the intermediate structure that is very important in order to inhibit the enzyme. In this way, we solve, to solve the problem, it's important to define a proper reaction coordinate. This can be easy when the reaction is not too complex. When we start to study reaction mechanism that involves many steps, we will start to build free energy surface. And this is a problem because we are basically limited by the three-dimensional space. And at some point, if our chemical reaction becomes too complex, we are not going to be able to follow the reaction easily, because then we are going to need to include an additional dimension for every collective variable involved, or for every bond that is formed or broken, for to say in some way. And that we call the course of dimensionality. But this problem was solved by the adaptive string method that was developed by Kirill Sinopia in our research group. This method solved this problem because the string reduces the number of reaction coordinates just to 1. But even if we are working with some, I don't know, with a not too complex reaction coordinate, we are going to need still to build the entire free energy surface, because we don't know how the reaction is occurring. We know the structure of the reactant and the structure of the products, but we still need to understand how this is happening. So in this way, if we want to evaluate the energy associated to this reaction, in which we need to form first this intermediate where the proton is being transferred to the amino group and the oxygen is performing the nucleophilic attack, we will need first to evaluate the proton transfer at a constant value of the reaction coordinate that is describing the nucleophilic attack. And then we need to repeat exactly the same step for each one of the steps involved during the other reaction coordinate. By doing this, we can obtain the total free energy surface associated to the chemical reaction. But as for every single circle is associated to a simulation. And this simulation can take the order of days in being completed. And as we need to run a high number of simulations, in this case, we are running 2,160 simulations, to finish one of these calculations can take between weeks and months. And on top of that, we are using this and an approximate level of theory. So we need to remember that we are only interested in the minimal free energy pathway. So everything that be outside of this path will be rejected. So that means that, for example, here we are rejecting the 98% of computational power. So for that reason, we need to use a different strategy. As I was mentioning before, this is the strategy that we are using nowadays. Here, we don't waste anything of the computational power that we invested to obtain the minimal free energy pathway. So because we are able to simulate only the region of interest. For example, here we propose an initial guess for the minimal free energy pathways in some space of collective variables. Basically, the string connects the region of reactant and products through a series of string nodes that are molecular dynamic simulation. The energy of the simulation is evaluated, and then, accordingly to this value, they are moved towards minimal values. In order to avoid to all of them to fall into its local minima, that are the structures of reactants and products, they are evenly distributed along the process. Once the string reaches its minimal free energy path, once the string converges, it converges to its minimal free energy path. And this path is used to define a reaction coordinate that describes the advance of the system. We use this converged path to define a path collective variable, which is a function that increases monotonically when the system moves along the path, and therefore this function can be used as a reaction coordinate for free energy calculations. So now we can see that by using this method, we decrease the number of simulations that we were using previously by 2 orders of magnitude. But this has, the main drawback of this methodology is that we need to run all those simulations at the same time. They must run in parallel. So it's here where Mare Nostrum starts to play a critical role in our research. First, because by using the adaptive stream methods, we decrease the number of simulations, and by using Mare Nostrum, we increase the quality of the simulation we were running. So now, we can now even improve the resolution of the calculation, because now we can even use more computational nodes. So let's talk about the results obtained using the adaptive stream methods and the superpower of Mare Nostrum. So as I mentioned before, there is a molecule that is being used already as a drug to stop the infection with the coronavirus 19. So in order, if we want to improve this and we want to propose a new inhibitor, we need to understand how this molecule is working. So when this molecule was presented, they also proposed a reaction mechanism in which they are assuming that the histidine that is in the catalytic diet is transferring directly the proton to the nitrogen group that is in the nitrile warhead in the inhibitor, and that the sulfur is already activated, is deprotonated, is performing the nucleophilic attack over that carbonyl. But when we run our preliminary classical molecular dynamics simulation, we saw that our water molecules get inside the active site. And we saw that it was highly likely that this water molecule will react, that will make part of the chemical reaction. So for that reason, we decided to test a different reaction mechanism than the one that was proposed initially for this inhibitor. So at top right, you can see a diagram that is showing the collective variables that we define in order to define the reaction coordinate. As you can see here, we are talking of 7 collective variables. Will be impossible to follow trying to use a free energy surface. For example, you can see that the collective variables 7 and 6 are describing the process to transfer the proton from the cysteine to the histidine. And the collective variables 6 and 5 are describing the proton transfer from the histidine to the amino group, to the water molecules once the histidine is being protonated. Collective variables 5, 4, 3, and 2 are describing the concerted process of the proton transfer from the water molecule to the nitrogen in the nitrile group. And finally, the collective variable 1 is describing the nucleophilic attack. So when we employ the string method to evaluate this chemical reaction, we saw that the first step was the activation of the catalytic diet. So it is not, this not exist in the neutral configuration as proposed, but it needs to be first activated. As you can see here at the bottom left, I'm showing you how the collective variables are changing while the reaction is moved through the reaction coordinate. You can see, for example, how the collective variables 7 and 6 are crossing. Every time you see these crossing lines in this kind of reaction, what you are seeing basically is that one atom is being transferred to the other one. So we obtain first a slow barrier energy for the formation of this ion pair structure. And from here, the reaction continues. And as we expected, the proton from the histidine is transferred to the water molecule, and the water molecule transfers its protons to the nitrogen in the nitric group. If you remember, we are interested in this value of energy because it is the transition, the energy of the transition state, which is related with the speed of the enzymatic chemical reaction. And also, you can see here that this is a highly concerted reaction, as all the events are happening at the same time. So we obtaining the barrier energy as we obtain a barrier energy that is related with the value of the catalytic constant that is reported experimentally. So basically, we can say that with this methodology, it served to us to validate the method. So, yeah. As we saw that the water molecule was important, and this water molecule will be involved during the chemical reaction, we decide to look for a molecule that contain chemical group that can mimic the role the water molecule was playing during the chemical reaction with the material bead. In this way, we found the hydroxymethyl ketone, which have this hydroxyl group that is placed basically in the same position where the water molecule was placed in the previous reaction. So as the reaction is basically the same, we define the same number of collective variables, so we are defining the same, a reaction coordinate is very similar to the previous one. So what we saw here, when the chemical reaction proceeds, we saw also the formation of ion pair structure, this time at a higher energy value than before. And here we are seeing that we are obtaining a transition state structure that is very similar to the one that we observed in the material bead. But the problem is that if you remember, that energy was 16.3 kilocalories per mole, and here now we are obtaining 19.7 kilocalories per mole. So the energy of for this reaction is too high. But why is the energy that high? Because we expect this will be lower as we already attach the hydroxyl group to the inhibitor. So by analyzing our molecular dynamic simulations, we saw that the distance between the oxygen that will receive the proton and the nitrogen, the one that will give the proton in the histidine group, they don't remain most of the time in the reactive configuration. That is the one that is between 2 and 4, because it's the distance the optimal distance between the oxygen and the nitrogen in order to transfer this proton. So then we realized that what is happening is that hydroxyl group has a big conformational freedom, so it's approaching and moving away from the nitrogen. So what we need to do is to stop this conformational freedom. In order to do that, we replace one hydrogen in the methylene group by a chloromethyl group. This bulky group, we thought, like, is heavy, so probably it will stop the rotation of this hydroxyl group. Then we analyzed the new molecular dynamic simulation that we run using this new compound, and what we saw is like the design proposal increased the distribution of the reactive configuration. So it was what we were expecting. But still, now we have another problem because we changed the molecule. Will the enzyme recognize this new molecule in the same way it was recognized in the first version? By using a technique that is called thermodynamic integration, we saw that yes, that is the case, that the enzymes will continue binding to this molecule. The inclusion of the chloromethyl substituent barely affects the binding energy. We saw that the energy doesn't change. If we were obtaining a higher value, a positive value, we were facing a problem because the new inhibitor will not be bind effectively to the enzyme. So by analyzing the molecular dynamic simulation on both inhibitors, we saw that both inhibitors contain the same interaction protein inhibitor pattern. As you can see in the x-axis are the P3, P2, P1, P1 prime groups that were the P groups in which we divide the inhibitor, and in the y-axis are the residues of the enzyme that are interacting with this inhibitor. What the colors mean? Basically, what we can see here is that darker the better. If the color is dark, that means that the enzyme is interacting strongly with this P group of the inhibitor. As the pattern interaction is almost the same, we need to center our attention in the main difference. We saw that the interaction of the version 2 of the inhibitor with the histidine 41 is increased, while the interaction of the version 1 of the interaction that the version 1 has with the asparagine 142 and the glycine 143 are decreased, as this hydroxyl group is not moving, so it cannot interact with them. But, okay, how is the inclusion of this chloromethyl group affecting the reactivity of the inhibitor? The result was overwhelming because the barrier energy is not just lower than the one that we obtained for the version 1 of the inhibitor, but this is even lower than the one for the Paxlovid. Now we obtained a barrier of 15.1 kilocalories per mole. In this way, the use of computational methods provide us insight for improving the design of antiviral drug against COVID-19. So we can conclude that the adaptive extreme method is a reliable methodology to be applied to develop covalent inhibitors, and that the inclusion of the chloromethyl group improved the speed of the inhibition of the reaction. Also that a strategy to improve the kinetic properties of the inhibitor was envisaged, and that the computational methods are a valuable tool in the drug design process. And finally, that by the use of high-performance computer, better levels of theory can be used to find more accurate solutions. I want to thank to the people in my team, especially to Iñaki Tuñón, my supervisor, to the Generalitat Valenciana for the funding, to the Barcelona Supercomputing Center and the Red Española de Supercomputación, and I want to thank especially to the people working in the support area. Thanks to them, I understood how to handle properly all the computational power that Mare Nostrum gives us, and thanks to all of you for your kind attention.
Are there any questions for Carlos? Yeah, you know, it's the last session, so we're going to start. But thank you very much, Carlos.
Thank you.
Thank you.